Okay, so that's a fancy machine, right? But how does this thing make ATP, right? In detail, how does this really work, right? We know that it has to somehow, but how, right? Now, the answer to this was first proposed, and the first model that sort of gave us some insight was proposed by another famous chemist who did a lot of work on helping us understand how this machine works, Paul Boyer. Um, and it came to be known as the binding change mechanism. And the way to understand this is to recognize a couple of things. The first is that getting a p-loop NTPase, of which the F1 is a member, to sort of bind to this pentacovalent intermediate transition state, which remember, if you remember, we talked about before in the hydrolysis of ATP, getting hydroxyl to attack that phosphate to generate this pentacovalent intermediate with all this negative charge, right? There's that water molecule coming in, right? This would dissociate. You get all this negative charge in that pentacovalent intermediate. That's a super high energy transition state. It's the reason that ATP hydrolysis has a high kinetic barrier. It's kinetically stable, even though thermodynamically, this thing really wants to split that guy off and create that new acid oxygen group and this free phosphate. Getting a p-loop NTPase, right, to bind this transition state and stabilize that transition state, that's not a hard thing to do, right? We sort of have thought about that sort of thing. That's what an enzyme catalyst is going to do, is going to recognize the structure, orientation, charge distribution, shape, whatever, bind this sucker really well and drop that transition state free energy barrier. And if you're going to hydrolyze ATP, right, we got that because the thermodynamic balance here is massively in the direction of ADP and organic phosphate. Question is, how do you go the other way? It's easy enough to get something to bind that transition state and get you to equilibrium faster, but how do you bias the energetics so now ATP is more stable than ADP and inorganic phosphate? Okay, well, this is Boyer's idea. And his hypothesis was that, okay, what's going to be the case is that the three of the subunits, and it turns out it's the beta subunits in F1, are going to exist in three possible conformations when this machine is active. Now, the alpha subunits also bind ATP, but they're catalytically inactive. They're just structural, right? They bind ATP, but they don't do anything with it. It's basically just a structural component. All the business is happening on the beta subunits. And Boyer said, or suggested, one way to make this work would be if the beta subunits populate three different conformational states. One he called O for open, right? And that's basically the state that's going to load inorganic phosphate and ADP and release ATP. Another called L, which you think of as kind of a closed state. It's got ADP and inorganic phosphate bound, but now it's not freely exchangeable with the free solution. And then the business subunit, which is the so-called T state. And this is the state that makes the ATP. And the idea here is that the T state does two things. One, it binds tightly that that pentacovalent transition state, dropping the free energy barrier to access that state. But it does something different compared to what you would normally think of. It binds ATP really tightly too, right? And when it binds ATP as tightly or maybe a little more tightly than ADP, inorganic phosphate, that binding now, rather than having, you know, ATP here and ADP here, it now equalizes the free energy between ATP and ADP, maybe even tips it in the direction, if you're making ATP, tips it in the direction of being slightly more stable in the T conformation to have ATP there than ADP. Now makes some sense, right? Because now if you can drop that transition state free energy barrier, and if you bias the energy by binding really tightly to the ATP in the direction of ATP, now you can make ATP. And it doesn't even have to be that big a bias, right? You can basically have this thing tipped just a bit and you're going to potentially make ATP and do it quickly if you can get that transition state free energy barrier down, right? That's the idea. But the trick is if you're binding the ATP really tightly enough to like shift that free energy balance, how do you ever let go of it, right? And the key to that is the mechanical motion, the actual physical mechanical work done by that gamma epsilon stalk as it rotates through the complex, right? That's what switches the conformation of the beta subunits between just binding ADP and making ATP and then letting go again, right? So this is the basic idea of the binding change mechanism. So here we've got looking down end on one of the F0 units, and there's the gamma stalk in the middle. Let's say we've got the green subunit over here. It's open, right? And it's just, you know, reloaded with ADP and inorganic phosphate. But this is sort of in this state. This is in equilibrium with free solution. It can load in and out. We got the L state where the subunit is now closed. The blue one is over here closed. So this is not in free exchange, right? It's bound in the active site and it's stuck in there. And now the gamma stalk, right, has positioned or has switched the conformation of the third beta subunit, which had ADP and organic phosphate bound. Now it's binding to the ATP tightly. It's dropped the transition state barrier, and now it's locking on to ATP, and it's pulling that equilibrium in this direction. So it actually can shift the energetic balance while bound much more into the ATP, right? Okay. The trick now is the asymmetry that we talked about at the interface between the gamma stalk and those three different subunits, right? The gamma stalk interacts with the beta subunit in three different ways, and that interaction and its position governs whether the subunit is in the O, the L, or the T state. All right, so here's our starting position, right? If we now rotate the gamma stalk one position around, and the orientation we're looking at it and synthesizing ATP, it would go in this direction, to go counterclockwise. That would then flip, right? Because remember, the 120 degrees around the position of the gamma stalk was the open position. That would now bring down here to the yellow subunit. That would push it into the open state. And remember, it was making ATP, so now this can now release that ATP it was making. But look what the conformational shift has done over here to the next beta subunit around the circle. It's flipped it into the T conformation. This one is now binding ATP tightly, binding the transition state, right? Dropping that free energy both at the transition state and stabilizing dramatically ATP relative to ADP and inorganic phosphate, and letting this thing now populate a significant amount of ATP by stabilizing and binding the ATP tightly. And now it's closed on the green subunit. It's closed the one that was loading from free solution, right? And it's closed that in, right? So now we rotate one more, right? So ATP can now come out of the open subunit, the yellow subunit over here, because we had moved that guy around, right? This guy, as I said, was now making ATP. This guy is now closed. This subunit, the open subunit, now can reload, right, with ADP and inorganic phosphate, right? And once that reloads, right, we're basically ready to go again, right? The subunit now, as a result of another round of movement, will eventually, it's going to swing on over here, right, which will then close this guy. This will go to the L conformation, right? This will then go to the O conformation, and this one will be at the T conformation. So you can see how that would work by rotary repositioning of the that asymmetric gamma stalk between the three beta subunits. Okay, like I said, this was Boyer's binding change mechanism, and it made a lot of sense. And there was a lot of accumulating evidence that suggested something like this seems like it had to be going on at some level. But this model also made a very specific physical prediction, right? It predicted that, and nobody had really shown, again, there was indirect evidence that seemed like this had somehow kind of maybe had to be going on, but no one had shown that the F1, F0 was a rotary motor, right? That the gamma stalk actually rotated by virtue of this, reaction. So that was sort of a missing piece, right, to the demonstration, right? Again, people kind of logically assumed something like this maybe had to be going on, but nobody had shown it. Until the 1990s, when a group in Japan led by Masasuke Yoshida did an incredibly clever and elegant little experiment. What they did was they did an optical single molecule microscopy experiment where they took some F1 and they put it down on a glass cover slip in a special microscope known as a total internal reflection fluorescence microscope, or a TIRF microscope. And what TIRF allows you to do is look at fluorescent objects in very close distances relative to, say, the glass substrate that you're essentially reflecting a laser beam off of. And you can look at things within a couple hundred nanometers of that surface. Now, if you had fluorescently labeled to try and see what was going on in the F1, you wouldn't see anything, you just see a spot, right? You're limited by diffraction. But what Yoshida did was something very clever. He realized he could make a covalent modification of the gamma stalk right at the tip, and he could couple to that an actin subunit. And actin, of course, as you hopefully remember from cell biology, makes these nice long polymers. And you can control that by how much actin you've got and capping proteins. So you can make these long filaments that are easily visible as sort of a line of fluorescence in a microscope that's otherwise limited by diffraction. And you covalently attach these, make the actin filament fluorescent. You can say, well, what happens if I now run this motor in reverse, right? If I have it make ATP, which the F1S0 can go both ways, right? Does it act like a rotary motor? And the answer is, yeah, it does. So that's basically an image from Yoshida's actual paper showing a single F1 motor sitting right there in the middle of that with an actin cable sticking off of it, supplied ATP, and he's driving it hydrolytically, and it is rotating, a direct physical prediction consistent with the binding change mechanism. Here's another one. So the other thing is, if you slow the motor down, you decrease the amount of ATP you're giving it. Prediction is that if you looked at where the subunit is, right, with time, the beta subunits are distributed around that F1 about every 120 degrees, right? So if you slow down the amount of ATP you're giving it, slow down hydrolysis, you should see that actin cable sort of stall at predictable angles, right? Around basically a circle of 120 degrees. And guess what? That's exactly what it does, right? So this is an actual data record of the stepping of the F0 in an experiment like this. And when you look at the spatial orientation within the microscope reference frame, you see these three like spots where the cable sort of stalls and then moves, stalls and then moves when you're at limiting ATP, right? So all of this basically is a beautiful demonstration that the F1 F0 really is working as a rotary motor, and the binding change mechanism is a simple way that rotary mechanical motion could be coupled to a change in conformational state that biases the free energy between ATP and ADP and inorganic phosphate, right? Which is cool. All right, well, the missing piece now is, that's great. How does proton movement make that happen? Like, nothing I've talked about up to this point explains how movement of a proton would make this thing spin, right? And that kind of, was a missing piece for a while. And the potential solution to this, or likely solution to this, came from suggestions made by Howard Berg and George Oster. It was partly a consequence of their studies of the bacterial flagellar motor and some things they saw in common potentially with some of these ideas that were bubbling up out of how the complex 5 F1F0 might work. And the secret comes down to a couple of things. The first is this contact point between that C ring of the F0 and that A subunit, right? That interface is really, really key, and some of the properties of that A subunit are really key. The other thing that's really key is an amino acid in the C subunits right there, right in the midplane of the bilayer. That amino acid, sort of highlighted in green there, that's an aspartic acid residue, right? And for those of you who remember Dr. Carr's lecture at this point, you should look at that and go, wait, what? That's on the outer face of the C ring, exposed to the bilayer, the hydrophobic bilayer, and it's an aspartic acid, right? Really polar charged. How is that even possible? How could that even be there, right? And the key to how this works is that changes in the ionization state and positioning that aspartic acid residue are directly the cause of or linked to um, uh, rotational movement of the C ring, right? And so here's the idea of how this works, right? So there's that C subunit again, right? And there's that aspartic acid residue sort of right smack in the middle of that C subunit helical pair. The other key piece to this, again, as I said, is that A subunit. And the idea here is that the A subunit has a channel. It's the channel through which the protons are going to move from the intermembrane space back into the matrix with the electrochemical gradient, but it's split. The channel is in sort of two halves. There's a half channel that's exposed to the cytoplasmic face or the intermembrane space, and then there's a half channel that's exposed to the matrix, right? And it's not continuous in a single channel. They're in two half pieces. And the idea here is that inflow of protons from the intermembrane space, from the outside, is going to be directly coupled to the ability of the ring to rotate, right? Okay, so here's the idea. Here is sort of the C subunit ring. Here is the A subunit. There's the two half channels. And in principle, it's possible to have an aspartic acid residue exposed to the membrane here if two conditions were met. One, it's protonated, all right? So that aspartic acid side chain, that carboxylate group is protonated. There's no hard charge. And those polar side chains, that carbonyl and that sort of OH group, if they're hydrogen bonding with something nearby, if that's sort of tucked away against a neighboring subunit and it's hydrogen bonding satisfied, you can neutralize and dramatically eliminate the polarity. That could now be moved into the bilayer. So if it's protonated and tucked away in just the right way, you could have this thing rotate into the bilayer, and that aspartic acid residue is not a problem. All right. So the under conditions where that side chain is deprotonated, right? Now we have sort of two spots here. We have two C subunits, two aspartic acid residues, one oriented towards the or positioned with the half channel into the matrix, the other positioned with the half channel into the intermembrane space. And if they're deprotonated, the thing's locked, right? By virtue of the fact that you're not going to push that negative charge into the bilayer, A. B, I'll show you in a minute, there's a really important hydrogen bond, really strong ionic/hydrogen bonding interaction between a key arginine residue and the A subunit on the sort of intermembrane space facing side of the ring, right? The combination of that deprotonated charge and that strong salt bridging ionic interaction locks the C subunit in place, right? Until something happens, it's not going to go anywhere, right? And that's something that happens, of course, you can probably see where this is going, is a proton diffusing into that channel, protonating then that aspartic acid residue, right? Which now provides the charge neutralization. It also results in a release of the hydrogen bonding salt bridging interaction with that key arginine residue and allows that subunit to get sort of tucked away with the polar group satisfied and more or less neutralized from the point of view of being able to be pushed into the bilayer when you bring that proton in. That essentially releases the lock and allows the motor to rotate one step, right? Bringing the other subunit, right, that was negatively charged now into position that locks in place with that arginine, right? But notice what happened. Notice the next subunit over that had a hydrogen bond, when it toggles one position over, that guy is now exposed to the matrix facing half channel, right? The solvent filled matrix facing half channel. That proton can now dissociate, right? That proton dissociation now gets us back to where we started. And you can see basically what's happening is that a proton coming in to the half channel that's facing the matrix, right, allows it to be neutralized, right, by protonating the aspartic acid residue, allow it to be tucked away. It then rotates around the circumference, or as the C ring rotates around, that proton then comes around eventually back over to the intermembrane, the matrix facing half channel, where it's then released. So the flow of the protons is happening comes in, goes around, and then goes through. And it's that flow of protons and the neutralization of that aspartic acid residue that gives the direct coupling of rotational motion to the flow of protons in. Okay, well, that's cool. I mean, that can explain how the thing can rotate, but that doesn't really explain where the force is coming from. Why does it rotate? Why? I mean, is it just that you neutralize and then it just sort of like it's flopping around and maybe it eventually toggles over? It's possible that that's kind of part of the mechanism, right? Though it's not obvious why that would happen very efficiently. It looks like there may be more to it than that. So in the structure that was solved of the A subunit with the C subunit, there's a really interesting feature, right? Here's that arginine residue I was just mentioning, and here's that that sort of intermembrane space, cytoplasmic facing half channel, and there's the aspartic acid residue, and there's that salt, really strong salt bridge / hydrogen bond interaction with that arginine residue and that deprotonated carboxylate of that aspartic acid residue, and they're really close together and they're just like tight, locked in place. But notice something else. Look at the orientation of this C subunit helix relative to these guys. You'll notice that when the aspartic acid residue engages that arginine, this helix sort of kinks and rotates to the side, right, in order to position that aspartic acid residue so that it can make that contact. So what's thought to happen is that when you reprotonate this aspartic acid residue and disrupt that interaction, that allows that helix, and there's a significant restoring force because the helix kind of doesn't want to be there, right? There are other stabilizing interactions that want to restore this helix back to this conformation, right? And it's more or less being held there by the interaction energy of that arginine and that deprotonated aspartic acid residue. When you protonate that guy, that basically like releases, you can think of it as sort of releasing a spring, but to reposition it, it has to push on the C subunit, right? That repositioning sort of pushes against the C subunit as it reorients. And when it pushes, the restoring force from that pushing force essentially pushes off the C subunit, right, in this direction and helps rotate it to the next position, right? Okay, so that's the idea, or current idea, about how proton flow through these two half channels. Protonating and deprotonating this key aspartic acid residue in the C ring is directly related to driven mechanical motion of the C ring, which moves the gamma epsilon stalk, forcing the beta subunits of the F1 to toggle between open, close, and high affinity for ATP, right? Okay, that's pretty cool, right? That puts a whole lot of these ideas together in one little nano machine that lets us synthesize ATP for all that work we were doing for oxidizing those carbohydrates and oxidizing those amino acids and other things that we can run through TCA cycle. So a couple of final points. One is that this sort of whole cascade is directly linked to the need for ATP, right? The F1, F0, as well as the rest of this cascade we've been talking about, if the ATP levels get too high, this machine goes off, right? Even in the presence of an energized mitochondria, it's not going to run, right? So there's a very tight coupling between ATP needs in the rest of the organism and the running of the oxidative phosphorylation cascade. And part of that is sensed by ADP, right? And the F1, F0 and the oxidative phosphorylation cascade is directly connected to the activity of a couple of antiporters. And one of these antiporters is important for getting ADP into the mitochondria. And when ADP is imported in, that antiports the ATP, the F1, F0 just made, back out into the cytoplasm, some of it anyway, right? There's also a hydroxyl phosphate antiporter that brings the inorganic phosphate in that's going to be needed to make ATP. That's an antiporter of hydroxyl, right? So flow through these channels is very tightly regulated with needs for ATP and activity down the electron transport chain and the activity of the F1 and F0, right? Okay, that's more or less where we finish, right? We're now sort of at the end. It's been a pleasure getting to teach you guys and sort of go on this little excursion through central biochemistry. One of the ways I like to end is with a sort of a final video where some of these guys in Cambridge and Massachusetts have used very high-end rendering computer sort of vision and molecular modeling to build these, really neat sort of videos of life in the cell. And I would normally show that, but I'm not going to do it on this video. But if you look at the slide stack that goes with this video that's posted, there's a link on this final slide right here, this powering the cell, that'll take you to the sort of final video that I generally like to use to finish the class, that goes through many of the different steps we've been looking at in detail, but in a nice sort of visual summary with all kinds of fancy graphics and the F1, F0 turning and ATPs coming in out of the mitochondria and electron transport. And you'll see that ATP glows, which is kind of cool. So with that, guys, thanks again and good luck.